Hello everyone, myself Mansi and my partner Drashti from 4th year Biomedical Sciences will be discussing about BLAST and different types in this video. So BLAST is an acronym for the Basic Local Alignment Search Tool and refers to a suite of programs used to generate alignments between a nucleotide or a protein sequence. Basically, it allows you to insert a sequence of nucleotides that you have obtained through a sequencing reaction from any database, and it will compare your entire database of known genes and will allow you to identify the gene which both protein encodes or RNA and which species it comes from. There are four main different types of blast. First is the nucleotide blast for the nucleotide sequences. Then there is blast X for the translated nucleotide sequences. Then there is T blast N for the protein and the protein blast for the protein sequences. First we'll see the nucleotide blast. For this we need to enter some nucleotide sequence that can be obtained from some database. So here we will be using the NCBI given by the NIH, that is the National Library of Medicine, and we'll search for some of the enzyme, like the most common one is the hexokinase. So for all of the database we'll choose the nucleotide as our database. By searching the enter button we will have different examples of the nucleotides. This all are the results of hexokinase. By clicking on the first one, that stands for Drosophila melanogaster, we will get its sequence. So here it represents its accession number, definition, the source and organism from it is collected. But here we are focused on collecting its nucleotide sequence. So we'll be directly going on to the FASTA. In bioinformatics and biochemistry, the FASTA format is a text-based format for representing either nucleotide sequence or the amino acid sequences, in which nucleotides or the amino acids are represented using the single letter codes. So this FASTA sequence can be directly copy pasted into the BLAST window, or else we can use the accession number. Now we will be copy pasting whole of the FASTA sequence in order to put into the BLAST window. Now going back to the BLAST site, I'll open the nucleotide BLAST window. A new window will appear which will allow us to enter our sequences. Here you can enter the accession number, the GI number or the FASTA sequence. Here we will be entering the FASTA sequence. Now this is a query subrange which stands for how many number of nucleotides has to be compared, but will not change any of the parameters. Here there is a job title which is a descriptive title for your BLAST search. Then there is a database. Generally we need to select the standard database that is inserted into NCBI. Then there is an organism if we are working on specific organisms like the mus musculus or the Rattus norvegicus. Then there is the entrance query. It tends to limit our search. Then the next is optimization or program. Generally, we will be using the Mega Blast for highly similar sequences. And now we can click on Blast in order to find our results. A new window will be opening and it will take some seconds in order to show and load the results. Like this. So there is a job title as we have done the nucleotide sequences, and the following information is seen. So coming to the descriptions, here there is a list of the organisms or the genes of the sequences that have been found similar to our entered sequences. So there are about different types of results which can be seen here. So there are different columns which stand for different significance. So here is the maximum score which says that how much percentage of entire sequences has been sourced through, and it could have different values ranging from 100%, 83%, 94%. The highest maximum score is suggested for 100%, which is the better. Then there is the percentage identity which depends on the E value. The percentage identity describes how similar the query is to to the aligned sequences. The higher the percentage identity, more is the significant match. Now the next is we can see is the graphic summary. So this will show the distribution of top 117 blast hits on the 100 subject sequences. So it is a graphical representation of our results. The color of the boxes represents the score of alignment, with the red representing the highest alignment scores. Generally, the higher the alignment score, the more is the significant hit. Then the next one is coming to alignments. The sequence alignment shows us how well our query sequence matches with the subject sequence in the database. The matching sequence consists of a combination of characters and the empty space. Now this line represents the identical basis between the query and the subject. In some of the cases, we can also find an empty space which can be represented by a dash character in either query or subject sequence, which denotes a gap in the alignment which is still not discovered or entered into the database. Then the last one is of taxonomy. So taxonomy report summarizes the relationships among all the organisms found in the BLAST results. Using this report, it is easy to see how many records are found within the broad taxonomic groups. So here we are seeing various results. Clicking on any one like the Drosophila organism, we can see its entire taxonomy, and it can be used for further studies.
The second type of BLAST is BLAST X, that is the translated nucleotide sequence searched against the protein. So, taking an example of hexokinase and selecting here is nucleotide, click on the search button. You can run BLAST from this page also. Click on the first one, that is Drosophila melanogaster hexo A. Here you can see the accession number, and on the right hand side there is a run BLAST, so click on that. We want BLAST X, so click on BLAST X, and the accession number is already copied here. Scroll down and click on the BLAST option. Basically, in BLAST X, a nucleotide sequence is used as query, which is first translocated in all 6 reading frames, and then each of the translated amino acid sequence is compared to the protein sequence in protein databases. Basically, this is recommended for identifying the protein coding genes in genomic DNA or cDNA, and also whether the novel nucleotide sequence is a protein coding gene or not. This might take some time. In the results, you can see on the left hand side there is given a job title, program, database, then query ID, description, query length, and molecular type. Scrolling down, here will be the description, graphic summary, alignments, and taxonomy. So as we discussed earlier, the graphic summary will show the red color if it is the highest hit, and accordingly it will continue. Then comes in alignment, where it shows how well our query sequence matches with the subject sequence in the databases, and the matching sequence consists of the combination of characters and the empty spaces. So this line will show the identical basis between the query and the subject sequences. And last one is the taxonomy. Also, in BLAST-X, the results will show the comparison between the level of amino acids, that is the aligned amino acid sequence, that is the translated query versus the homologous sequence in the protein databases. The next type of BLAST is T-BLAST-N, that is the protein query searched against the translated nucleotide. So, taking an example here of albumin, and selecting category as protein, as we are using the protein query, and then click on the search. Select the first one or any of your interest. Then click on Run BLAST. Here also all the categories are as same as previous ones, and then click on the BLAST. This T-BLAST compares the protein query sequence to all the 6 reading frames of the database, and so it is widely used to associate the protein with chromosomes or mRNA. In the results will be obtained on the new page, and here you can see that amino acid sequence is used as a query to compare it with the translated nucleotide, that is coding sequence, which means the amino acid sequence is compared at the protein level with each subject nucleotide sequence translated in all 6 reading frames. Here you can see that descriptions, graphic summary, alignments, whatever I have told right now, and the taxonomy. Now taking an example of protein keratin. Here selecting again as protein. Click on the search option. Select any of them. I'm selecting here as keratin partial for Homo sapiens and Run BLAST. The parameters will remain same here also. So this BLAST-P, that is the protein BLAST, is a database where the protein sequence is searched against the protein sequence itself, and it is used for both identifying a query amino acid sequence as well as for finding the similar or homolog sequence in the protein databases. The results are of similar kind, having descriptions, graphic summary, their alignments, and taxonomy.